Доброго дня, шановні колеги. Ви підключаєтеся до вебінару Центру громадського здоров'я. Наразі ми чекаємо,
Hello, dear
доки
colleagues.
всі
You're
наші
now
запрошені
joining the
спікери
webinar
доєднаються,
organized
і
by the
тому
Ukrainian
буквально через
Public Health
декілька
Center.
хвилин будемо
We're now
починати наш
waiting for another
сьогоднішній
minute
вебінар.
to make sure that all the participants who were willing to join can actually join our meeting. And then we're going to make a good start in about a minute or so. Thank you very much.
Ще раз всіх вітаю. Я бачу, що до нас доєдналося
Hello, everyone.
вже
Once
майже
again,
200 слухачів
I can
сьогодні.
see we almost have now around 200 participants
Сьогоднішній
joining
вебінар
our
буде
webinar. So
присвячений
I suggest we
тематиці
start.
визначення
Today
клінічних
we will
основних
speak
механізмів
about
протимікробної
identification of
резистентності
clinical
у
important
грам-позитивних
mechanisms
бактерій.
of
Сьогоднішня
antimicrobial
лекція
resistance
є
in
продовженням
gram-positive
циклу
bacteria. Our
вебінарів
today's meeting is a
виробництво-вирощування
continuation
інфекційного
of the
контролю
European
та
experience
адміністрування антимікробних препаратів перспективи
series
для України. Цей
and
цикл
also for
вебінарів
the
ми
sake of
проводимо за
expanding
підтримки
Ukrainian
наших партнерів, про
experiences
яких
in
ми
the field of
знаємо
AMR.
далі вже
We
з
conduct this webinar
2023
series
року.
and we
І
will speak
всі
about
наведені
the
матеріали є в
supporters and our
відкритому
partners
доступі на
a bit later
YouTube-каналі
who
Центру
support us
громадського
a lot
здоров'я
about
ми з
that. But please
України,
be
і ви
aware
можете
that all
їх
the
завжди
materials,
передивитися
these
та
are video
ознайомити
recorded and
свої
they're going to
знання
be
щодо
available on the
вищенаведених тематик.
website
Вже
of the
були
Ukrainian
попередні
Public Health Center and
вичерпні
you will be able to look
питання,
into
що
that once
стосуються
you need that.
інфекційного
Before we
контролю,
spoke about infections
епідеміології,
control,
адміністрування
epidemiological
антимікробних
surveillance,
препаратів, догляд за
antimicrobial stewardship.
інфекціями та
We
інші
talked
питання,
about
що
taking
стосуються
care of
інфекційного
your
контролю.
medical devices
І
and all
хотілося б
other
окремо
health
висловити
related
вдячність
topics. I would
колегам та
like to express
партнерам,
my
які
deep
дозволяють
gratitude
та
to our partners
допомагають
and
нам з
colleagues who make
організації
it
цих
possible
заходів.
for us to
Це
organize and conduct these
організація
meetings.
Центру контролю та
This is
профілактики хвороб Сполучених Штатів Америки,
the
ICAP
American
при Колумбійському
CDC Center,
університеті та
ICOP
досвідчені
at the
фахівці,
Columbian
колеги
University
з
colleagues, and
ЕСКМІД,
also
міжнародної
well-experienced
організації,
colleagues
яка
from
надає
the
підтримку та ділиться
ASCMID
своєю експертизою.
European
На нашому
community,
заході
which are always very supportive for
передбачено
us.
переклад,
During
отож
our
ви
webinar,
можете,
we
якщо
have
є бажання
simultaneous translation
слухати
option
лекцію в
available.
оригіналі, а
So
ви
once you
можете
and
включити
if you want to listen to
налаштування.
that in the original, please
Вони
do
зараз
so.
наведені на
Or
екрані,
if you feel
є,
like
і таким
comfortable,
чином
you can
слухати
go to
український
the
переклад
Ukrainian
вебінару,
channel
який буде презентуватися далі.
following the given
Також хотілося
instructions on the
б
slide
проговорити,
and
що
just
цей
make
цикл
yourself
вебінарів
feel
буде
comfortable.
продовжуватися.
Let me also
Отож, ви
reassure
не
you, dear colleagues, that the
пропустите
given webinar
наступні
series will
лекції.
continue. So in order
Підписуйтесь на
to
Фейсбук-канал
not
Центру громадського
skip
здоров'я ми
these
з
meetings,
України, де
please
ми
go
завжди
to the
своєчасно
website
публікуємо
and
анонси нових
sign
заходів.
in
Підписуйтесь на
to our
YouTube-канал Центру
website.
громадського здоров'я, де ви
Also sign
будете
into our
бачити
YouTube channel of
записи
the
цих
Ukrainian Public
вебінарів,
Health
і
Center.
цей
You will be
вебінар
able to
також буде
watch
викладений
these
на
video recordings
YouTube-каналі.
as well
І
as the given
також,
webinar of
наскільки
today.
буде дозволяти
It will
час, тому
also be
що
available
по
on
регламенту
YouTube.
сьогоднішньої
And as
зустрічі ще одна
time
година.
allows, because we are,
Доктор
pretty strict with
Барбара
our agenda, it is going to
Holzknecht
be only 1
допоможе та
hour.
надасть
But
відповідь на
we
ваше
have
питання, щоб
Dr.
поставити
Barbara
ваше питання. Будь
Holzknecht, who
ласка,
is going to
пишіть
be with us
вчасно.
today. And if we don't have
Отож,
enough time for
власне,
questions
будемо
and
вже
answers, we
починати.
will be able to
Хотілося
deliver
би
these questions later to Barbara.
презентувати нашого сьогоднішнього
So
спікера.
maybe this is the due
Сьогоднішнім
time
нашим
to introduce
гостем,
you,
досвідченим
dear colleagues,
фахівцем
to our
буде
speaker.
доктор
So we have
Барбара
Dr.
Холцкнехт.
Barbara
Вона
Holzknecht.
старший консультант
She is a
лікарні
senior
Herlev і
consultant of
Gentofte
Herlev
при
and
Копенгагенському
Gentofte
університеті
Hospital at the
і є
Copenhagen
фахівцем Національної
University.
референс-лабораторії
And
з
she is also a
антибіотикорезистентності
specialist of the National Reference Laboratory for Antimicrobial Resistance in the Danish Serum Institute, and she will speak today about identification of clinically important mechanisms of resistance in gram-positive bacteria. So, Dr. Holzknecht, the floor is yours, please.
Thank you very much, and thank you for this introduction, and thank you for inviting me. So the topic for today is the detection of clinically important resistance mechanism in gram-positive bacteria. And apart from working both at one of the Copenhagen university hospitals and at the reference lab, as you have already introduced me to, thank you. I'm also affiliated to the European Committee on AST, on Antimicrobial Susceptibility Testing, UCAST, as the educational officer. So next slide, please. The agenda for today, I have selected some acquired resistance mechanisms I want to talk about, and in bold font here you can see the ones I want to spend more time about, because I think they are mostly clinically relevant, whereas the other ones I will not spend as much time on, and we will see whether we have time for the last one. So we will cover first of all methicillin resistance in Staphylococcus aureus. Then we will shortly talk about penicillin susceptibility in Staph aureus, then about glycopeptide resistance in Staph aureus, and then we will move on to VRE, vancomycin resistant Enterococcus species with a focus on Faecalis and Faecium. Then we'll move on to Streptococcus pneumoniae, where we will cover beta-lactam resistance. And then we will probably also have time to cover MLS, macrolide, lincosamide, and streptomycin resistance in Staphylococcus and Streptococcus species. Next click, please. So when I'm presenting, I will present the UCAST recommendations for testing these organisms. And the next slide, please. So we will start with methicillin resistance in Staphylococcus aureus. Next click. As you all know, the acronym MRSA stands for methicillin resistant Staphylococcus aureus. And so I think you have to click here 3 times. Thank you. And the clinical relevance of MRSA is, of course, that Staphylococcus aureus is one of the main clinical, the main human pathogens spanning infectious diseases as skin and soft tissue infections up to sepsis and endocarditis, bloodstream infections. And methicillin resistance has a great impact. The mortality of Staph aureus infections is much higher for MRSA as compared to MSSA, methicillin susceptible Staphylococcus aureus. Next slide, please. The genetic background is quite simple. Please, one more click. It's the MEG-A gene, which is located on the staphylococcal cassette chromosome, the SCC-MEG. About 15 years ago, another gene, the MEG-C gene, another variant has been described. It's not as prevalent as MEG-A, but it also exists. And then the next click, these 2 genes then code for an altered penicillin binding protein, the PBP. In the case of MEG-A, it's 2A. In the case of MEG-C, it's 2C. And these altered penicillin binding protein has decreased affinity to beta-lactams, and that, please click once again, is confers resistance to all beta-lactams. So penicillins, cephalosporins, except the 2 which have been designed to treat MRSA infections, ceftaroline and ceftobiprole, and then the carbapenems. The prevalence of methicillin resistant in Staphylococcus is different in different geographic regions. Please, the next slide. Here you can see the MRSA epidemiology. So this is data from the CSAR surveillance, WHO European region data from 2021, and showing data on the percentage of invasive Staphylococcus aureus isolates which are resistant to methicillin. And the color code here is that the dark blue represents the lowest percentage, less than 1%, and then getting lighter blue over light brown into dark brown, you get higher prevalences, with the highest prevalence shown here on the map being the dark brown, which refers to 25 to 50%. And you can see that there is a north-south gradient in the prevalence of MRSA, with lower prevalences in Northern Europe and higher prevalences in Southern Europe. So the next slide, please. How do we test phenotypically for MRSA susceptibility? Well, we do that by screening with cefoxitin. And if you click once again, then you can see the UCAST, one slide from UCAST from the homepage, displaying the performance of the cefoxitin disc containing 30 micrograms cefoxitin versus the MEG-A status. So this is aggregated data from 11 published studies, altogether 2,300 isolates approximately, and about half of them being MRSA, being MEG-A positive. There were no MEG-C positives in these in this collection, and you can see that the red isolates, the MEG-A positive MRSA isolates, are very well separated from the MSSA isolates by the cefoxitin disc. So the red isolates, the MRSA isolates, all have, or nearly all have, cefoxitin inhibition zones of 21 or lower, 21 millimeters or lower. Please, the next click. So what we have just seen is a very nice separation of the wild type, the isolates without acquired resistance versus the MRSA isolates. And this is then, so this is the UCAST recommendation that you do screening with cefoxitin, preferably by disc diffusion, and you use the cutoff of S susceptible is equal or greater than 25 millimeters, and resistant isolates will have zone diameters of less than 22 millimeters. And in gray here, just to mention that for other Staphylococcus species, the detection of methicillin resistance will be a little bit different, and you will have to go to the UCAST tables to find these recommendations. And then the next click, there are two expert rules. The first one saying if the isolate, if a Staph aureus isolate is resistant in the cefoxitin screening test, then you should report it resistant to all beta-lactams, except ceftaroline and ceftobiprole, the ones which are designed to treat infections caused by MRSA. In that case, these have to be tested individually. And the second expert rule, please click again, is that if an isolate is susceptible in the cefoxitin screening test, then you can report as susceptible to all beta-lactams which have recognized anti-staphylococcal activities. This will be the ones with notes in the UCAST table. Let's move on to the next slide. As I have shown you with the distribution of the cefoxitin zones, the cefoxitin disk diffusion test is very good to discriminate the MRSA from the MSSA population. But if you choose to confirm genotypically or with antigen testing, there are possibilities. And the first one being, please, the next click, the NAAT, the nucleic acid amplification test, so PCR or PCR-related technologies where you can test for the resistance gene MAC-A or MAC-C on pure isolates. And just a small note here, if you want to use a direct PCR test on clinical samples, for example, on screening samples, and these are samples where you can also have coagulase negative staphylococci, then you have to add a test for the junction of the SCC-MAC cassette into the Staph aureus genome, because otherwise you could get a positive MAC-A, MAC-C result from just normal skin flora, not being in Staph aureus. There is a latex agglutination test also available. Please, the next click. And that detects the altered penicillin-binding protein 2A. It's a test which is which performs very well and is easy to perform, and can be an alternative. You just have to be careful or to consider that it does not detect PBP2C. So MAC-C positive isolates would be false negative in this test. With that, let's go over to the next topic, which is penicillin susceptibility in Staph aureus. Thank you, and you can do another click. Just to say that penicillin susceptibility testing is clinically important because benzyl penicillin is the increasingly preferred treatment. There's a large multi-center trial going on Staph aureus bacteremias in Australia and other countries, and the first results have been presented at this year's ASCMED Global conference in Vienna, also showing that benzyl penicillin probably is the most relevant treatment for PSSA, penicillin susceptible Staphylococcus aureus. The next click, please. The molecular background is also quite easy. It's one gene, the BLAZ gene, which encodes penicillinase, which is a serine beta-lactamase. And the epidemiology actually is, in contrast to other resistance mechanisms or resistance prevalences, shows an increasing susceptibility. If you click once again, we will see the data from the Sentry program, so international program collecting clinical isolates from both the Americas, Europe, and Asia. And you can see here the percentage of MSSA, methicillin susceptible Staph aureus, which are penicillin susceptible. And this is rising from 14% in 1997 to 2000, the start of the study period, to 26% in 2013 to 2016. If we go to the next slide, we can shortly look at how to test phenotypically for penicillin susceptibility in Staph aureus. And one more click, then we get some UCAS distribution pictures here. I think you have to click once again. Then we see the criteria when you may report a Staph aureus isolate as penicillin susceptible. And that requires two things. Both you have to have a large inhibition zone, larger or equal to the breakpoint of 26 millimeters. And then also you have to inspect the zone edge, and that has to show the beach phenomenon. So a decrease and growth towards the zone edge. On the other hand, if you either have a zone which is less than 26 millimeters, or you don't have the beach phenomenon, then you have to report as resistance. So if you click once again, we see the pictures here. We call that the cliff phenomenon, where you have an abrupt end of growth at the zone edge. So both inhibition zone diameter and zone edge are important to consider when reporting benzyl penicillin susceptibility in Staphylococcus aureus. If you click once again, this is beyond the UCAST recommendations. Just to tell you the Danish experiences, we have been testing Staph aureus for benzyl penicillin susceptibility for a long time using the cloverleaf test. So the principle being here that you have a penicillin susceptible strain on your agar in the bottom, and then you make a cross with your test isolate, and you put a benzyl penicillin disc in the middle. And if you see the formation of a clover leaf, as you see here on the right-hand side picture, that means that your test isolate produces a beta-lactamase and thereby then allows growth of the underlying PSSA isolate, and that then gives you the formation of the cloverleaf test. You can read about the performance of the UCAST distribution method and what the cloverleaf test can give you on top of that in this publication I have cited here. Another way to confirm susceptibility is PCR or PCR-related technology. But the UCAST diffusion test is performing very, very well, so that alone can also stand, but you have to combine both the inhibition zone and the zone edge. So let's move forward to the next topic, that is glycopeptide resistance in Staphylococcus aureus. And I have only one slide for that because it is a very rare phenomenon, but it's of course important to know about. If you do one more click, then the first phenomenon I want to mention are the vancomycin resistant Staph aureus. So that is when A mediated high-level resistance, vancomycin MICs higher than 8 milligrams per liter. This is easy to detect, but it's a very rare phenomenon. So that has only been described in very few cases around the world. And well, luckily, this is a very rare instance, although we have many instances where we have Ben A VRE, which we are going to talk about in a minute, and Staph aureus in the same patient, and still the transfer of Ven A from an Enterococcus to Staph aureus occurs very, very, very rarely. And this is not difficult to detect. If you do one more click, the next phenomenon is the vancomycin intermediate Staph aureus. So these are Staph aureus isolates displaying vancomycin MICs, which are elevated 4 to 8, but not high resistant. And the mechanism for that is a thickening of the bacterial cell wall, which binds vancomycin. And these isolates will be detected by MIC determination, but preferably by broth microdilution. Another phenomenon related to visa is the heterovisa, the heterogeneous visa. And if you click once again, so that describes the phenomenon where you have a subpopulation of a Staph aureus isolate, which is vancomycin, displaying vancomycin elevated MICs of more than 2 milligram per liters. And the detection of heterovisa is very cumbersome, and is something which has to be done at a reference laboratory because it requires the PAP AUC, the population analysis profile investigation, which to be fully open here, I have never performed. So that is the principle is that you grow the isolate on different acres with different vancomycin concentrations and then plot it in a curve. But that is something which has a place in the reference laboratory. There are screening methods building on the principle of using a high inoculum because you want to detect a small subpopulation. And these are, for example, the macrogradient test and stacoplan and screening agar. But altogether, these phenomena are not very often seen, quite rare. And if you click once again, most visa and heterovisa are MSA, and you can have that in mind when you design your testing strategy. And just to tell you, in our country, for example, in Denmark, we have done sentinel surveillance on a national basis of MRSA isolates where we have not found visa or heterovisa. And for example, that has led to that in our lab, we do not test vancomycin susceptibility in Staphylococcus aureus. We would only do that, or we only do that if there is treatment failure. Let's move forward to the next species, Enterococcus species, and vancomycin resistance. So VRE, vancomycin resistant enterococci, will appear if you click once or 3 times more. And from a clinical and infection control point of view, clearly the most important species here are Enterococcus faecalis and Enterococcus faecium. If you click once more, that will appear also on the screen. The clinical importance of vancomycin resistance in enterococci is, and especially in Enterococcus faecium, which usually has high rates of acquired ampicillin resistance, is that then there are very, very few treatment possibilities left because enterococci are born with many resistance mechanisms already. Faecium, most of them have acquired ampicillin resistance, and then if you don't have vancomycin for treatment, you only have very few options left. Let's move on to the next slide, where we see the molecular background. And if you click once more, we will see that it's the molecular basis is it's a gene complex with genes referring to the transposition, and then a regulatory element, and then the VAN-H, VAN-A, VAN-X genes really being responsible for the glycopeptide resistance. And if you click once more, we can see on this little cartoon that the result, you can see on the top of it, the regulatory process, and then the end result being a target modification where the cell wall precursor, the end of the pentapeptide has one amino acid substituted. If you click once more, you can see that it goes from a D-ALA to a D-ALA DLAC, and that means that you have a target modification, an altered vancomycin binding site, which has lower affinity. If we then go to the next slide, we will see the VRE epidemiology. Same principle as we did for the MRSA map. So again, this is data from 2021 from the CSER surveillance. And you can see here that the pattern is a bit more patchy. There's no really gradient. And if you would dig down into the data, you would see that there are even within one country also quite large regional differences. We go to the next slide. This is an overview from an article in 2008, but I still think this is the best table to give an overview over how complicated it can get with VRE genes. So we have acquired resistance genes when A, B, but also D, E, G, L. And then we also have intrinsic resistance. So the Van C being a part of the chromosome and Enterococcus gallinarum and Enterococcus calyflavus. But that is not as much as a treatment problem, as many of them will be ampicillin susceptible, and it's definitely not an infectious control problem with regards to spread in hospitals. At least in the hospital I have been working at and what I'm aware of. So by far the most important here are Van A and Van B. And if we take one click more, we can see that Van A gene confers high level resistance to vancomycin and also resistance to teicoplanin. So it's easy to detect. The only thing which can be difficult about Van A is a phenomenon which is called VVE, vancomycin variable enterococci. And that means that they are Van A positive, but vancomycin susceptible. However, these isolates will convert to a vancomycin resistant phenotype if you expose them for vancomycin either in vitro or in a patient. So they should be treated as VRE isolates. The second mechanism, Van B, if you do one click more, confers low level resistance to vancomycin, can also be high, but can be low. And these isolates will be teicoplanin susceptible. So they can be difficult to detect because they can display low level vancomycin resistance. And they are called low MIC Van B if they have very low MICs and even being into with MIC levels of the wild type. So both VVE and low MIC Van B can only be detected by molecular testing. But that is, of course, very resource heavy, and in a non-outbreak situation, I do not think that is something which should be mandatory. So it really depends on whether you have VVE clones in your area, whether that would be relevant or not. Otherwise, I just wanted to mention it, and I don't think that this is to put the area to put your resources in. Let's go to the next slide where we see a part of the UCAST table on vancomycin susceptibility testing on enterococci. And you can see here that for Enterococcus faecalis and faecium, the MIC breakpoint is 4 and the disdiffusion breakpoint is 12. But there is also a note, and if we do one more click or two more clicks, we can see that it's not only important to take the zone diameter into account, but also, again, to inspect the zone edge. And here it's the sharp zone edge we want to see to be able to report or isolate as vancomycin susceptible. So that's the picture A. Whereas if you have something looking like pictures B, C, or D, so having fuzzy zone edges or colonies within the zone, then you have to either report them resistant or proceed with confirmatory testing. So the pictures B and C are quite typical Van B isolates. So again, here it's important to both take into account the zone diameter the inhibition zone diameter and the zone edge. If you perform MIC testing, I want to draw your attention to a ucast warning. If you do one more click, you can see that there is a ucast warning active on vancomycin susceptibility testing on Enterococcus faecium and faecalis using gradient tests, because gradient tests can underestimate the MIC, so you can get false susceptible results. Let's move on to the next slide on phenotypic and genotypic susceptibility testing. Well, wrap up and the expert rules. So if you click once more, we can see the first expert rule, which refers to the VEN-B phenotype. So if an Enterococcus isolate is vancomycin resistant and teicoplanin susceptible, the VEN-B phenotype, then you should report with a warning of resistance development to teicoplanin. And that is because there is some data showing that teicoplanin resistance can develop under treatment. So VEN-B isolates should not be reported teicoplanin susceptible without a warning. If you click once more, then we get another expert rule which refers to the vancomycin variable phenotype, or no, not the phenotype, the vancomycin variable enterococci. So if you have an Enterococcus isolate which is phenotypically vancomycin susceptible, but VEN-A is detected by molecular methods, then you should report it resistant to vancomycin and teicoplanin. And then one expert rule referring to the low MIC VEN-B. So if you have got an isolate which has such a low MIC of vancomycin that it's in the susceptible range, but you have detected VEN-B, for example, because you have inspected the zone edge, then you should report that resistant to vancomycin and had a warning of resistance development to teicoplanin during therapy. One more slide about VRE, about the genotype. If you want to perform VEN-A or VEN-B PCR directly on a clinical specimen, again, that could be relevant for a rectal screening swab, for example, then VEN-A PCR will have a high specificity for detection of VRE. And that is because VEN-A has nearly only been described in Enterococcus species, whereas VEN-B is present in other bacterial species. So a direct PCR has low specificity for the detection of VRE. With that, we move forward to the next species, the Streptococcus pneumoniae, pneumococci, and beta-lactam resistance. And there, let's first have a look at the MIC distribution of benzyl penicillin and Streptococcus pneumoniae. If you click once, we will see the picture. So this is from the UCAST database, where you can find all kinds of MIC distributions, always in this format, where you have the twofold dilution steps of the MIC on the x-axis, and on the y-axis, you have the percentage of microorganisms tested in this column diagram. And on the left-hand side, with the lower MICs, of course, you will see in blue the wild type. If we click once again, we'll see the animation. The wild type population is the population which has not acquired resistance. And the highest MIC of the wild type, in this case 0.06 milligram per liters, is the epidemiological cutoff, the ECOF. Then on the right-hand side, if you click once more in red, we see the population with acquired resistance. And here the resistance mechanism is a modification of the PBPs, the penicillin binding proteins, which then have a lower affinity for beta-lactams. And that is actually because of horizontal gene transfer from Streptococcus viridans group to commensals to Streptomoniae, leading to mosaic genes. So there is really a continuous there's not only high level resistance, there is a development of different degrees of resistance, which you also can see here in the graph. This is clinically important to detect this resistance, both whether the isolate is within the wild type or not, and then how high the level of resistance is. If you do one more click, we'll see that it's clinically important for treatment of meningitis and also endocarditis, which is of course a lot more rare than meningitis for pneumococci, and that is because non-wild type isolates, so isolates with an MIC of more than 0.06 milligram per liters, are not treatable with benzyl-penicillin. And that is translated into clinical breakpoints, which are susceptible S is less than or equal to an MIC of 0.06, and everything above 0.06 should be reported as R if the isolate is from a meningitis or endocarditis. Whereas for other infections, for example, pneumonia, benzyl-penicillin in higher doses can be used for treatment of isolates with an MIC of up to 1. So there the clinical breakpoints are S susceptible is equal or less than 0.06. Then for the MIC 0.125 to 1, that is the I category, meaning susceptible increased exposure, so high dose, and isolates with an MIC of more than 1 will not be treatable and should be reported as R resistant. If you do one more click to the next slide, we can see the epidemiology of pneumonia, the percent the prevalence. Next slide, please. Going to the susceptibility testing, and this is actually the only topic here of all the topics I have covered where there has been a recent type isolates. That's from that has been the case for many years. But the second point here is new. If you have an isolate which is oxalin screening positive, so displaying an inhibition zone of less than 20 millimeters, you can use the benzyl-penicillin 1 unit disc to differentiate whether it's a benzyl-penicillin I versus R. And that is, of course, for non-meningitis endocarditis isolates, because these should be reported R in any case if they are oxacillin screening positive. So no need to perform MIC testing, but you can solve that with a benzyl-penicillin disc. If we go to the next slide, we will go step by step through the testing diagram. We start with the oxacillin disc, 1 microgram. And if you already have a benzyl-penicillin also on your first plate, that will allow you to report already on the first day all isolates, but you will not have a look at the benzyl-penicillin disc for all isolates. So we start with the oxacillin disc. And yes, you can see here that if the oxacillin zone is more or equal to 20 millimeters, then you can report the isolate susceptible to all beta-lactam agents with clinical breakpoints or a note. And there is one exception there, but you don't have to do any further testing. If we then go one click further, we can see if we have screening positive oxacillin isolate, so on a zone diameter of less than 20 millimeters, then we have detected beta-lactam resistance, and we can already report all meningitis and endocarditis isolates as resistant to benzyl penicillin. And then we can have a look at the benzyl penicillin disk, and if the zone is more than or equal to 14, then we can report it as I, susceptible increased exposure, and the patient can be treated for example, pneumonia with high-dose benzyl penicillin. If the zone is less than 14, it should be reported resistant for benzyl penicillin. And then for other beta-lactam agents, if we do one more click, we have a look again at the oxacillin zone. And if that is between 9 and 19, there are quite many agents we already can report susceptible. The aminopenicillins, piperacillin, cefepime, cefotaxime, ceftaroline, so the third generation cephalosporins and carbapenems, imipenem, and meropenem. And only for beta-lactams, which are not included in this list, you have to perform testing of the agent in question, and that is the next click here. So let's move on to the next click, and that is just to draw your attention to a UCAST warning. You can see here on the slide that apart from oxacillin disc diffusion screening, you can also use benzyl penicillin MICs, but you should not use gradient tests for that. That should be broth microdilution MICs. So there is a warning from UCAST that benzyl penicillin gradient tests do underestimate the benzyl penicillin MIC in pneumococci. In many occasions, in all our labs, we would use the oxacillin disc to do the screening. Let's move on to the next slide and the last topic. So that is MLS, macrolide lincosamid streptogrammin resistance in Staphylococcus species and Streptococcus species. And if you go to the next slide, we'll see that for testing, we use the macrolide erythromycin, and clindamycin is the only lincosamid, and we also use that for testing. There is one important resistance mechanism which is conferred by the gene and leads to a methylation of the ribosomal binding site. So again, a target modification, and that comes in a constitutive phenotype and an inducible phenotype. And you have to test that by placing an erythromycin and a clindamycin disc next to each other. And if you do 2 more clicks to get the animation going, you can see that you have to place these 2 discs edge to edge with certain millimeters apart, which differ for Staphylococcus and Streptococcus species. And you can see on the pictures below, this is constitutive resistance, where you have growth into the clindamycin disc. You have a zone diameter of 6. We would read this here. Whereas if we click once again, we see the inducible phenotype, where you have a deformation of the clindamycin zone to the side where you have the erythromycin disc. And that is called the D phenomenon because it looks like a D. If we do one more click, we'll see that animated as well. So that's a positive D test. There are other resistance mechanisms as important here. So if you do one more click, that is an efflux pump, the MSR gene, which gives the erythromycin or clindamycin S phenotype. And if you do one more click, that is without a positive D test. And then there is also another resistance mechanism giving the phenotype macrolide S and clindamycin R, or also S, which is a lot more rare. But that was just to explain the D-test phenomenon. And if we go to the next slide, we can shortly talk about the clinical relevance of that. And that is because if you see the inducible resistance, there's usually a small subpopulation which is constitutively resistant. And that's why UCAST recommends to report them as clindamycin R. However, you can include a comment for Staphylococcus species and the hemolytic streptococci saying, sorry, you have to click once more, saying clindamycin may still be used for short-term therapy of less serious skin and soft tissue infections, as constitutive resistance is unlikely to develop during such therapy. But the point here being that if you have a serious infection, especially with a high inoculum, a prolonged therapy, you can see that constitutive resistance will develop during therapy. And if you want to do some further reading, you can see that when you click again in this reference. And one more click, and we will be at the last slide here. Just to say that UCAST has a guideline for the detection of resistance mechanism, which you can find at the homepage. This is currently under revision. If you click once more, we will see that also on the screen, but I expect that for the gram-positives, probably there will not be too many revisions, except for the Streptococcus pneumoniae. And with that, that was my last slide, and I want to thank you for your attention, and we open the floor for questions.
Дуже дякую за чудову лекцію.
Thank you very much for your wonderful lecture. And we would like to express our gratitude to you for joining us and for sharing your experience, since it's very useful for our bacteriologists. And we have a few questions. The first question is, it's rather a request, if you could just share your experience that all inpatient, not all inpatient facilities have bacteriological labs which use UCAST methodology. If you could share in what way the patient's materials are sent to the lab. What are the arrangements for that?
Well, in our little country where ways are short, distances are short, we also have not a lab in each hospital, but we, for example, our lab is serving 4 hospitals, and we will then ship the, we have arranged transports with cars selecting samples at the biochemistry laboratories at the hospitals who don't have a clinical microbiology department, and they will then drive with these samples several times a day to the hospital where the laboratory is. I hope that was answering your question.
Дуже дякую.
Yes, thank you. As far as I understood, also colleagues were asking about the methodology used by a bacteriological lab, because in Ukraine, the laboratories are supposed to use UCAST methodologies. What about Denmark?
Yeah,
Is it the same?
Yeah, we all use UCAST. We have 10 departments in Denmark and all use UCAST methodology. The vast majority of tests being disk diffusion tests. So UCAST disk diffusion methodology is the main method used for AST, for antimicrobial susceptibility testing in our country.
Дуже дякую за відповідь. Наступне питання.
Thank you for your answer. The next question is the following. Is it possible to detect or suspect VVE enterococci if a lab does not use a molecular method?
Well, the short answer is no. In our region, we have had an endemic clone of VVE, so we have had to set up molecular testing of all Enterococcus faecium isolates, and we are now considering going back because that is a very that is taking very many resources. No, there is no good way to do it without a molecular test. And I think if you don't, but it will be very rare that you have VVE in your hospital. So if you want to do something, I would recommend some sort of surveillance of invasive isolates, for example. And if you then detect something, then you can think about something else. But if I would implement it, I would start with the invasive isolates, and that can of course also be done not real time, but run in batches, pooling samples. If you want to have some kind of surveillance with regular in regular periods to see whether you have VVE or not. But it is, I know that other Nordic countries have been performing molecular tests on invasive isolates for a long time without detecting any. So it is not, it is a rare phenomenon, and unless you're unlucky to pick one clone which is spreading in your hospitals, it will be a rare phenomenon and a very resource heavy thing to detect. So I do not think that this is where you should put your main resources. I think there are many other things which are more important in susceptibility testing.
Дякую за таку розгорнуту та практичну відповідь.
Thank you very much for very practical answer. I'm sure that was what our colleagues wanted to know. And the next question is the following. Is it possible to detect susceptibility to cephalosporins in pneumococci by using a gradient method?
I would advise against it because you can solve it with the oxacillin test for the vast majority of the isolates. So then the oxacillin, you can infer resistance from and susceptibility from the oxacillin test. If you have the oxacillin test, that's only one disk, you can report a whole bunch of all relevant beta-lactam agents without doing any further testing. So that will spare you a lot of resources and work.
Дякую за відповідь. Колеги, враховуючи,
Thank you very
що
much for your answer. Dear colleagues, since we're a little bit short of time, I suggest we have two final questions to our today's speaker, and we will then complete today's mission. And the next question is, in your opinion, can we say that the MIC method and DDT method are comparable?
Yeah, that is basically what UCAST is doing all the time when breakpoints are set. So each time breakpoints are set, there is UCAST looks at the association between the inhibition zones with a UCAST distribution methodology and MICs, and looking at reference MICs, so broth microdilution MICs. So this is there is a lot of data behind the inhibition zones, the inhibition zone breakpoints, and that's why in our country we rely on the distribution testing, which is where you need less. It's less expensive, and it's a standardized method, and whenever you use an MIC testing method, which is not the reference method, not broth microdilution, you will first have to see how that performs, and often we don't have too much data for that.
Дуже дякую за відповідь. Я би хотів ще
Thank you very much for your answer. I would ask some additional question just to clarify this, because the Ukrainian Public Health Center in their counseling work, and in particular our bacteriologists, point out certain moments. So we would like just to separate MIC and these diffusion methods. In terms of determining susceptibility to certain antibiotics like to colistin, we understand that it can be unpredictable, it can be unforeseen by this diffusion method. So if we want to test susceptibility of such antibiotics as vancomycin, etc., they are not supposed to be in place.
Well, thank you very much for that. And probably the last question. I see the last question on the chat box. The calculation of MIC dose, some kind of calculations or some calculators for that. How does it look like on your side?
Let me just go back to the last question, and just to say that I endorse your comment on that. That of course there are some exceptions for the distribution, but then you will not find any UCAST breakpoints for the distribution. So of course for each test you're setting up, you have to look into the table whether there are exceptions or whether there are notes. So my comment was just on a general note. To the last question here about calculation of MIC dose, I'm not sure I entirely understand. What I think you ask about is using MICs for therapeutic drug monitoring, and then relate the isolates MIC to the drug concentration in the patient. If that is the case, then my answer would be you have to be aware that the MIC you get, that there are some that there is a uncertainty or a range. An MIC is never totally correct. If you saw the wild type distribution I showed you for benzyl penicillin and pneumococci, you saw that the wild type really spreads about 5 twofold dilution steps, and within the wild type, with the methods we have available, we cannot differentiate between, for example, now talking benzyl penicillin and Streptococcus pneumoniae, we cannot differentiate between an isolate which has an MIC of 0.06, 0.03, and the ones lower than that. So we can only say if we perform an MRC testing and whether it's 0.06, 0.03 or lower, it will be within the wild type. So you should pay attention that you don't use the absolute MRC in any calculations calculating drug exposure in a specific patient, because an MSC will not be or not always be exact, and that is very true when they are in the wild type, and otherwise you should at least say ±1 dilution step. I hope I answered your question. Otherwise, please have a follow-up question.
Можливо, колеги, які задавали питання, зможуть прокоментувати, тому
Those
що
who asked the question, maybe you could comment on that, because I personally can hardly comment on that. That is not my topic, frankly speaking, not my cup of tea. Maybe this is a nuance of translation, but probably you were speaking about the potential correction of the therapeutic dose based on the MIC data. Can different MIC value impact our choice of dosage? If I got it correctly.
Yeah, and the short answer, black and white answer to that would be that in UCAST we have two susceptibility categories, one referring to the normal dose and one referring to the high dose. So the S means susceptible if you use normal dose, and you can go to the UCAST tables which dose that should be. You can always go higher, but you shouldn't go lower than that, because the breakpoints are based on a certain dose. And then the I category is susceptible. Increased exposure means that you have to treat the patient with high dose, and again that you can see in the UCAST table.
Дякую. Насправді стало набагато більш зрозуміло, і тому що
Thank you very much for that. Thank you very much for that. That made it much clearer, because that really adds to your understanding if it is susceptible or if it is susceptible under a higher dose. So there is susceptibility retained, but you need to modify the dosing regimen, meaning you have to increase the dose of the antibiotic. Thank you very much, Dr. Holzknecht. I can see lots of words of gratitude on the chat box. They thank you very much for the session, for the lecture, for this Q&A session. We have exhausted our time, and with that, I think I will say from American CDC Center, AskMed colleagues. I would like to thank our interpreters today, and also to you, Barbara. So guys, I wish you have a nice day today and the peaceful skies above. And tomorrow you will have this video on the website of Ukrainian Public Health Center. So please look for other meetings and other webinars on our Facebook page as well. All the best.
Thank you very much.